Okay, so my presentation is going about computational modeling of the role of epigenetics in anxiety disorders. Um, yeah, I've chosen this subject for my thesis. I'm a third year's bachelor in computer science, and uh, yeah, so I'm going to present you briefly but concisely my paper and what I've done in it. So what am I going to talk about in this presentation? I'm going to present you the model I've used for the role of epigenetics in anxiety disorders, um, also the functions I've used. Then I'm going to show some simulations that I've done. And also I'm going to propose future research for my paper and for this subject. And I'm also going to introduce you about anxiety disorders, what it means and what includes. Okay, so anxiety disorder, what is anxiety disorder exactly? Anxiety disorder is, or patients experience uncontrollable, persistent, and excessive fear or worry. And this is not the same as innate anxiety or worry that most of the people encounter in their daily lives. For example, being anxious before a big exam or a big presentation is very normal. This is just nature's mechanism, and this is a healthy mechanism. But patients um, experience the same uncontrollable, not the same, but uncontrollable and excessive fear or worry when there is even not immediate threat or danger. And it also affects their lives on a daily basis. So that's the difference between patients with anxiety disorder and normal fear or anxiety. So there are several factors that cause anxiety disorder. There are chronic stress, for example, but also emotional or physical abuse, and past or childhood experiences. And I've chosen chronic stress as factor for my paper because um, there has been done research recently in 2023. There's been released a paper which includes or talks about stress-induced anxiety. So um, yeah, there's my paper also based on. So that's the reason I've chosen chronic stress as factor for my model. And the subject also includes epigenetics, so the role of epigenetics on anxiety disorder. And what are epigenetics exactly? So there's a difference between genetical disorders and diseases and epigenetical disorder and diseases. So genetical diseases include mutation in DNA, and most of the times they are irreversible disorders or diseases. For example, the syndrome of Down. And epigenetical diseases or disorders, there is a problem in the functioning of the gene. So the expression of the gene is not working properly. So a gene could be turned off or turned on. That's the difference. And most of the times, those epigenetical diseases or disorders are reversible. So what could influence a epigenetical disorder? For example, epigenetical factors as, as environmental stimuli, stress, for example, but also diets. So diabetes is an epigenetical disease um, based on, for example, someone's diet. And that is then, I'm talking about diabetes too. So in the paper of the researchers my paper is based on, discussed a certain microRNA, which is in this case specific miR4A35B. And this element X, yeah, I call it epigenetic. There's no official name, but yeah, we can take that name for now. It acts as an intermediary between environmental stimuli and the expression of genes. So this is between chronic stress now and the anxiety gene, which is in this case, researchers have found PGAP2, which is responsible for the anxiety patient experiences. Okay, then we have, then I'm going to show what is the difference between a healthy mechanism and the mechanism someone has with anxiety disorder, but this I'm going to talk about later in the simulations I'm going to show. Okay, so which models have I created for this project? I've created a model about the development of anxiety disorder. So here we are going to see that a normal person develops anxiety disorder and what are the different stages of this and what are the roles of the underlying mechanisms in the brain and the amygdala, etc. And also I've proposed a therapy to heal anxiety disorder. So which functions have I used? I have used the advanced logistic combination function. The formula for this function is here. And the parameters for this function are the steepness, sigma, and tau for the threshold. And these are used to, these are used when there are multiple connections in a node. I'm going to show the nodes and the model after this slide. And this function is important for the case I've created, which is the development of anxiety disorder. And also I've used the step once function, which uses the parameters for the start time and the end time. And this function I've used to manipulate the environmental stimuli. And in this case, that is chronic stress and stress. No, chronic stress and therapy. So you're going to see that in the simulation as well. So this is the model I've used. So here you can see multiple levels. So this is a higher order adaptive self-modeling network. Here we can see the base level. This base level is used from Professor Trur's book. And this base level indicates emotion regulation. So you can see some context states, and these context states, in my case, are environmental stimuli. So here we can see context state for stress, context states for chronic stress, and they both begin to impact the first stage, which is the sensing state, so the sensory state. Then it goes to the sensory representation state, and this is about the stress, so the feeling of the stress. Then it goes to the preparing state of the emotion, which is, in my case, anxiety. And all these black arrows are positive connections, so they have a positive impact. And the dotted lines are negative impacts. So here we see also then the feeling state of anxiety, which is the same as the amygdala in the brain. And here we have the PFC, so the prefrontal cortex, and this is the controlling state. So yeah, this state is trying to control the anxiety the person experiences. Then you have these blue arrows, which are negative impacts on the most upper W state. And what are these W states? These W states are self-model states that represent connection rates so that the network can model itself concerning the connectivity. So this state, this blue state, represents the brain structure. Then we have this state that represents the enzyme that is responsible for anxiety. Then we have the messenger RNA, the yellow level. Then we have the DNA, and in my case, we have the PGA-P2. And then we have the above level, the most upper level, which represents the genetic level. And that is the first one which is impacted from the feeling of stress. So we can see here, when a person feels stress, then finally the preparing state, the feeling state are impacting the epigenetic level. The research that has been done talks about stress-induced anxiety. It means that when the person feels stress, the normal mechanism, the healthy mechanism, is upregulating the microRNA. And this microRNA in this level is trying to suppress the anxiety gene, which is PGAP2, so that the person does not feel excessive anxiety. Further, we have these pink arrows, and these are just the impacts from the W states. And as we can see, the mechanism consists of 5 self-adaptive modeling levels. And this is the first model for the development of anxiety. And then I have also created a second model for my project, which consists of just a new context state, which is context therapy. And this state acts as an intravenous injection of an element microRNA-4A35P. And this state has a positive impact on the W state of the epigenetics. And this therapy has a strong positive connection, because the researchers have found that microRNA has the most significant role of suppressing anxiety in a person. So that state should help to recover the healthy mechanisms in a person to suppress anxiety. Okay, so then I also have created some simulations in MATLAB with software of Professor Treur. And for the following scenarios, so the development of anxiety disorder and the treatment of anxiety disorder. So here we have the scenario which I've gotten from MATLAB. Okay, so firstly, we can see that so this line at level 0.5 represents the context stress state. So this is stress a person can experience, and this activates everything. So the mechanism, the biological mechanism. So what happens? The sensory state is going up, so it's sensing stress. That is the same for sensory representation state, but finally it becomes stable here as well. So that this is still a healthy mechanism. And we can see that the feeling state, so the purple one and the yellow one, so the preparing state, are also going a little bit up, but then remain stable. And this is pretty normal. So the person is feeling a little bit anxiety, and that's it. As well for the prefrontal cortex, which is trying to suppress, of course, the anxiety, and yeah, it is going up, so it has a positive It is still working. Then we know from the model I've shown that the healthy mechanisms should go up. So here we see the yellow going first up. So that is this one. So this is on the epigenetic level for the microRNA miR4A35P is going up, as well as the underlying W states. So this is a healthy mechanism because here the body is trying to suppress the anxiety, which is good. But then from around timestamp 120, with the step 1's function, I have manipulated the chronic stress. So that is the brown line to go up to level 1. So that is from here, it's going up. Because as I have said, chronic stress is one of the most significant factors for anxiety disorder. And here we can see then that the preparing state as well as the feeling state are going up. But now, firstly, this sensory state and the sensory representation state are going very up because the stress is more and more. Then we can see, because of this, that the preparing state in yellow and the feeling state are going up as well. So the person is feeling more anxiety right now, which is normal. But then also the prefrontal cortex is going up. So this means that the prefrontal cortex is trying to suppress, to regulate this emotion that is felt by the amygdala until here. Because here, the prefrontal cortex cannot regulate the emotion anymore because of the high levels this experiencing. So the impact from the feeling state and the preparing state are felt by the upper mechanisms. So we can see that the yellow line is going down from the upper W state. So the microRNA is not being able to work properly, and this is going down, and the rest follows. So that means that finally as well, as we can see here, that the feeling state is going as well to a very high level because the anxiety cannot be suppressed anymore. So the feeling state is going up, and the preparing state as well. And we can see that the prefrontal cortex level is going down until it reaches level 0, which means that the body is not able to handle any anxiety more and to control the anxiety. So we can see that now the patient has developed anxiety disorder. How can we confirm this? You can see that even if around 280, the chronic stress is not present anymore, you can see that the feeling state and the preparing state are still very high levels and remain constant. So now the body is not able to handle anxiety anymore, which is the case in patients with anxiety disorder. So then I have scenario 2. So this is the scenario where I have presented the intravenous injection of the microRNA, which should regulate the expression of the anxiety gene, PGAP2. So as you can see, here is what I've described in the last slide. So here we see, okay, the patient has anxiety disorder, which is not regulated anymore. But then with the green line, so that is the therapy line, the intravenous injection, it is going up around 460. You can see that the mechanisms are going up again. So these are the yellow one is the mechanism from the microRNA level. And the underlying mechanisms are following. And what happens? The prefrontal cortex, the green line here, is going up to regulate. So that is working again. And we can see that the preparing state, the yellow one here, as well as the feeling state are going down. So because of the healthy mechanisms are going to work again, that begins from the most upper stage which represents the microRNA mirror for H35P. Now the anxiety feeling is going down. And what you can see is that the prefrontal cortex now is remaining stable but positive. And the feeling state is stable here as well, stable. And how to confirm that the patient now has no anxiety disorder anymore? I've manipulated the therapy, so now it's off from around 6:20, the timestamp. You can see that the feeling state, as well as the prefrontal cortex state, as well as the prepared state, are remaining pretty low. And this is normal because we still have the normal stress the person is experiencing, which is this blue line. So here we can see that the healthy mechanisms are restored. Okay, so the conclusion is that normal stress activates the healthy mechanism, and this is an innate mechanism which is already since the beginning of human beings. And we can see that chronic stress disrupts this healthy mechanisms and shuts it completely down. And the therapy I have proposed refers the disorder. As I've said, epigenetical diseases or disorders are reversible. And we've seen in the last slide that the mechanism is going, is restored, is being restored. So it's brought back to normal. So for future research, other researchers can look into this therapy and find if it's feasible to do this intravenous injection, because the specific element, the microRNA for H5P, should not be too expensive, but also it's a question, is it feasible in terms of uniqueness? So does every patient have to get a specific microRNA which is compatible with his body or not? So that is a question for future research. And another type of therapy would be to upregulate, to increase the production of the microRNA. So that is different than injecting the microRNA. And if this is done, then the model should have another level above the existing model, because of the microRNA being upregulated, being produced more, then it should be a known underlying mechanism and not just a context state to add microRNA. Okay, so this is the end of my presentation about my paper.